Good evening, everyone. Thank you for joining us tonight for our RiverWatch Winter Speaker Series. For those of you who are unfamiliar with the Illinois RiverWatch Network, RiverWatch is a citizen science organization that trains volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. If you are interested in becoming a citizen scientist, please consider coming to one of our spring workshops that will be held in person across Illinois. Registration is now open and workshops are starting to fill up, so sign up soon to reserve your spot. Before we get started, I ask that you keep your microphone muted during the presentation to limit the number of distractions. We will have a Q&A session after the presentation, so if you have a question along the way, you can either put it into the chat box and we'll answer it at the end of the presentation, or you can save the question and ask it aloud. And with that, I would like to welcome Jodi Vandermyde. Jodi received her master's degree in zoology from Southern Illinois University. Her current position is Environmental Protection Specialist at the Illinois Environmental Protection Agency. Her work focuses on monitoring water quality and aquatic biota in Illinois wadeable streams. Tonight, Jodi will be talking about the Intensive Basin Survey Program.
Okay, thank you. Okay. Does everybody see that okay? Hopefully.
Yeah, you're good.
Okay. So welcome. Thank you for coming. I'm Jodi Vandermyde. Just to give you a visual of where I'm at in the EPA, I work in the Bureau of Water, the Division of Water Pollution Control, Surface Water Section in the Southern Monitoring Unit. There are 3 monitoring units in Illinois. There's the Northern Monitoring Unit located in Des Plaines, the Central Monitoring Unit in Springfield, and the Central Monitoring Unit in Marion, which is where I'm located. The red lines on the map roughly show the boundaries of where each unit samples. These units focus on monitoring streams and lakes in Illinois and assessing their water quality for our biannual integrated reports. We do this largely through 2 programs. One's the Ambient Water Quality Monitoring Network. This is where we sample fixed locations every 6 weeks, and our Intensive Basin Survey Program, which is what I'm going to talk about today. So just a general overview of today's talk. And before I get into the details of the intensive program, I'm going to discuss the importance of this work, give a little background on the program, how we select our sites, the need for reconnaissance before we sample. Then I'll go over our sampling methods for water inverts, habitat surveys, and the DNR fish surveys. Some details on our sampling processing and data management, and then our assessment process and finished reports. So why is this important? Why are we collecting physical and biological data to assess water quality in streams and lakes? And today I'm just focusing on streams. So the physical data can indicate habitat degradation, water chemistry issues, and the biological often reflects the physical data. So we can see this by looking at macroinvertebrate communities. Different invert adaptations and life cycles can tell you a lot about a stream. For some inverts, they need to breathe with delicate gills, like shown on this mayfly, that are on their abdomen. They need flowing water and stable substrate like rocks. Other invertebrates can live in more stagnant water because they use a siphon to directly pull oxygen from the atmosphere, like this mosquito larvae shown with the siphon just right at the surface of the water. As part of the Clean Water Act, when we assess our streams, we use criteria to determine if the stream is fully supporting or not supporting of its designated use. Majority of the waters in Illinois have a designated use. So what are these? We have the Clean Water Act has 5 designated uses, 5 different ones. This includes aquatic life use. So this includes aquatic organisms, but it also can include terrestrial, anything that lives around water, and also aesthetic qualities. So if the streams are pleasing to you, Recreational use, this includes canoeing, fishing, sailing, or anything really to do with water. And primary contact, so like swimming and fish consumption. So getting into the intensive basin survey, they are called intensive because they are intensive. We collect water, habitat, macroinvertebrates, and fish. This is done on a 5-year basin rotation, shown by the map on the right. So in 2020, the light blue watersheds were sampled. The darker green watersheds were sampled last year. And for this year, we're sampling the lighter green watersheds. Streams are selected within these basins. They need to be wadeable and accessible to us. The sample period is from June 1st to October 15th. We need to sample them at baseline conditions. Results are then used for our biannual water quality assessments, and then these are published in our integrated reports. So I'm going to sample or focus on what we're sampling this year, which is the lower Kaskaskia and the Little Wabash River when I explain our site selection process. So when we're selecting our sites, we look at past sampling history and make a note of the time that it was last sampled. This helps us get an idea of what sites have been recently monitored and what sites need attention, especially if the site hasn't been visited in over 10 years. Some sites are sampled every basin rotation, and this could be because we use them as a reference site or they continue to have multiple violations. So after we get our sampling history, we also look at the number of violations a stream has had. The map is highlighting stream segments with the number of violations from our previous reported 303D lists. And for example, I have, this is the lower Little Wabash. It has 3 violations. This is the CO1 segment, and it has mercury, total phosphorus, and dissolved oxygen flagged as violations. So after we've mapped sampling history and violations, we reach out to DNR to see if they require any sites to survey. This could include streams with recent fish kills, or they need to collect fish tissue data for consumption analysis. We also consider any special projects that may be happening in the watershed. This could be related to facility surveys. And finally, we look up past recorded notes on specific sites because, you know, there might be a reason why it hasn't been sampled over 20 years. This could be due to poor access, or maybe the landowner is not willing to grant us access. Just focusing on the Little Wabash River, when we're considering all the information, we can propose some sample sites and we can plot them on our map, and we can look for data gaps. So here, hold on, this is a new trick that I learned. This section here looks like we might be a data gap, so we might need to look into that closer, see if we actually need to sample something there, if it's important. Then we meet up with the DNR to discuss the proposed sites, and then we narrow them down for a reasonable amount to go for reconnaissance. So reconnaissance usually takes around early May. The 2 main reasons we do reconnaissance: first, we check to see if the stream is wadeable, and if we'll have any issues with obstacles like log debris jams or steep slick banks. The second thing that we look at the stream is if we can actually access it. We meet up with DNR to recon the sites. And if we can access the stream easily and safely. A few things that we look for is parking space for like 3 to 4 vehicles, a way to access the stream, especially for DNR because they have more equipment than EPA. They need to consider if they can get a trailer to the stream easily, as shown in this picture. to offload all their equipment. Also, if it appears to be on private land, we must try to gain landowner permission to park and have access to the stream. We do this by either knocking on doors or locating ownership, land ownership through county databases, and hope that the landowner still has a landline. It's become more challenging with people getting rid of landlines and using cell phones. So sometimes we have to mail a letter to see if we can have permission to, for access. So after all the proposed sites have been visited and agreed upon by EPA and DNR, we have landowner permission, we can start sampling. So the first thing we do is we collect water. Our field season again starts in June, and we need to collect 3 round water at base flow conditions. So we try to space these out to get a broad view of the stream, and we do this by timing our sampling in June, August, and October. The 2 main ways we collect water is by sampling on the bridge and by using a weighted bottle sampler shown here. Or by weighting the stream and collecting it by hand. Both ways we use a churn, which I'll explain further on the next slide. We use a new plastic half-gallon that you see in the weighted sampler for each site. And we use the plastic bottle, collection bottle, to fill our plastic sample bottles. And then we also use a glass amber to fill our glass sample bottles. We also take field readings using a multi-parameter probe, which is calibrated in the lab before we head out to sample. This includes water temperature, dissolved oxygen, dissolved oxygen saturation, conductivity, and pH. Water is generally collected facing upstream. Sometimes we have to collect downstream side of the bridge, especially if there's logistical or safety issues concerned. That's the only time we really sample downstream. If the stream is small, all the water sampling is done in the center of flow of the stream. 3 half-gallons are collected and placed in a churn. This is what a churn is. We collect in a churn to homogenize the sample before filling our bottles. And as the stream increases in size, we follow an equal width increment protocol. So medium-sized streams like the Big Muddy or Cache River, we collect water from the left, right, and center of the stream. And then large rivers like the downstream end of Kaskaskia or Mississippi, we collect 5 equal width increments. And I've also listed some parameters here that we analyze. This includes nutrients, metals, suspended solids, chlorophyll, TOCs, chloride, and fluoride. We also filter some sample water on site. You can see a pump here with a tube running from the sample water through the filter, and that we collect dissolved nutrients and dissolved metals in those. Once the bottles are filled, they are placed on ice, and we ship them to the central lab in Springfield. All EPA samples are processed and analyzed in the same lab. Excuse me a moment, take a drink of water. So when we actually get to a site, the first thing we do is take a habitat survey. And this is, we use the Quality Habitat Evaluation Index, or also known as QHEI. This gives scientists a quantitative assessment of physical characteristics of a sampled stream, similar to the index of biology integrity of biological data. The QHEI represents a measure of in-stream geography. So we get to the site and we first set our habitat reach before we begin our survey. The stream reach is usually 300 feet, or it's 20 times the width of the stream. So for example, the stream is 15 feet, we sample at least 450 feet. This obviously can be altered, especially if there's safety concerns such as impassable deep pools or any other sort of obstacle. So when we get to the site and we're got a reach set and we're walking the stream, we record what habitat is available. And the first thing to look at is macro habitats, which includes runs. This is shallower spots with some flow, riffles, which are shallow spots where rocks break the surface of the water, and pools, which are deeper and they lack flow. And we record this as a percentage relevant to the stream length or the reach length that we're doing. And surface area is taken into consideration. Other stream characteristics that we record, we get an average stream width. We get an average channel width. So if you imagine the stream is at bank flow, that'd be the channel width. An average depth and a max depth. The dominant stream substrate types, such as sand, gravel, or boulders. Any backwater areas or undercut banks. And then we also make a note of what's in the riparian area. If it's forested, if it's developed land, urban, anything like that. So after we have our notes, sites, the data can be entered either on site, as you've seen here, sitting on a bucket, working on the computer. Sometimes it's handy to do it on site, just so if you forget something while you're walking the stream, you can just look up and make a note. Or it could be done back in the office with notes. And then we always include a site sketch. So with our habitat information, we can allocate our 20 jabs. This is how we collect our macroinvertebrates. So the 20 habitat allocation is based on stream width. So if a stream is less than 30 feet, we take 12 jabs in the bottom zone and 8 jabs in the bank zone. So that's a small stream. This bottom right picture, we would take 12 in the bottom zone, 8 in the bank zone. Versus if the stream was larger, say between the 30 and 60 feet, we would take 14 jabs in the bottom zone and then 6 jabs in the bank zone. And that increases, if it increases to over 60 feet, then the bank zone decreases and the bottom zone decreases. So what are these? Okay, bank zone. Bank zone habitat has 3 different habitats. First one's overhanging terrestrial veg. This includes down trees in the stream or overhanging grass vegetation. Root habitat, so any tree roots that are submerged in the water, and woody debris. And if you can't tell, there are a couple caddisflies hanging on that log. There's one here, one there, one there. There are also the bottom zone habitats. The more common habitats are fines. This is sand or silt, and coarse habitats or coarse substrates. This ranges from gravel to boulders and sometimes bedrock. Generally more sensitive and tolerant taxa live underneath rocks, like this stonefly hanging out on this rock, especially if there's flow. So next time you're on a stream, if you feel like it, flip over some rocks and see if you find anything. The other bottom zone habitats is detritus. Detritus can be found leaf packs in some flow, or could be deposited along the bank. In aquatic vegetation, this could be macrophytes or filamentous algae. All right, so now that you know the habitats, we can start sampling some macroinvertebrates. We always try to work downstream to upstream. This avoids disturbing any habitat and prevents turbidity. If there's flow in the stream, it's not much of an issue, but a lot of streams, may not have flow, so then it's difficult to see habitat if you're working the opposite direction. So clear water just makes it easier to see the available habitat. And each habitat we discussed requires a little different technique, but it also depends on flow. In flowing water, you can see myself here sampling in some flowing water. We like to set the net downstream and then dislodge. I'm using my hand there, about a square foot in front of the net. That dislodged debris and inverts flow into the net. It's useful for quick swimmers like this megalopteran here, this Corydalis, and maybe some smaller mayflies that might get missed through the jab method. This can be used for all habitat types, only if the stream is shallow enough for the net. If the stream is flowing over the net, there's potential to lose the individuals, over top of the net. In slow water or deep pools, the net is jabbed 3 times in the same spot to capture debris and inverts. So if you can imagine taking a sample in some fine substrate, you would dip the net in the water and bump along the bottom of the stream, scoop it up, and then dip your net back in the water and bump along 3 times again. This helps kind of loosen stuff up in the sediment, and then you swoop in to collect the water afterwards. For roots in a deep pool, I like to scoop them up in the net, so the net's just above the surface of the water, and then shake them vigorously to dislodge any individuals. A lot of odonates like to hang out in roots, so they can cling on. So it's good to shake well. Also, we pick up a lot of rocks and wood, and we wash them in a tub. Caddisflies, shown here, they're tucked in this little crevice. Can't stick to the rocks. Same with like leeches and black flies. They're adapted to staying on the rocks and the wood, so some scraping and picking is required to dislodge them. After jab is collected in the net, it's deposited in a tub, seen here on the, for rinsing and sorting. We use a sieve bucket, shown here. It's basically a bucket with a mesh bottom that we collect all our jabs in, and then a green screen we have on top that's a larger millimeter mesh that can sort out the larger debris such as rock, leaves, and woody debris. This just makes our lab sample processing much easier later on. Clean stream water is added to the tub, and the contents are suspended and then poured into the bucket shown in the middle here. Organic matter, including inverts, float, so only the inorganic matter stays left behind. You can see some rocks and sand staying in the corner while the organic matter is flowing into the bucket. And this process is repeated until the water flows clear and no more inverts are seen in the tub. Some inverts, stick to the tub, so like snails and leeches, or if they're heavy like clams or rock caddisflies with rock cases, they'll have to be picked out individually from the tub and placed in the bucket. Most individuals are smaller than 1 millimeter, so they should wash through the screen and into the bucket. But there are larger inverts, such as these dragonflies and this giant water bug. Won't go through the screen, so they'll have to be picked and placed into the bucket separately. We don't survey for mussels. We only record if they're present or absent. If they are present, we make a note of what condition we find them in. So whether they're shells, relic shells, live or juvenile. We sometimes see the mussels as we're conducting our habitat survey. They can either be on the edge of the stream, or if the water is shallow and clear enough, we can see mussel trails. Here in the middle is showing a mussel trail, if you can follow it on the screen. If we didn't see any obvious signs of mussels, we briefly can feel along the bank and with our hands sifting through the silt and sand to see if we can find any. To wrap up the field sampling, it's not all fun and games. There are some sampling challenges. We do come across some, friendly neighbors that come and investigate and ask us questions and wants to know what we're going on. Some less friendly neighbors that interrupt us and disturb us for a while. With COVID, we had to, put up with wearing masks in extreme heat situations. And then, some days are just rough and it's hard to walk up steep cement banks, on mud. But overall, we manage. Just to briefly go over to DNR surveys. Generally, we use the same stream reach, but there are some logistical constraints that may prevent this, such as poor access, high conductivity, deep pools, or large log debris dams, like you can see here on the right. This would cause inefficiency in their sampling techniques. It's also possible they might have to do extra sites to collect fish tissue for consumption analysis. But also sometimes DNR and EPA do a site trade-off where DNR will sample a large river by boat that's difficult for the EPA to wade, and then EPA then can sample smaller streams that maybe the DNR has a hard time accessing to get all their equipment in. So this can result in more coverage for the watershed. It's better to have both fish and invert data for the site for the assessment, but at least some data or some biological data is good. So their most common method is using an electric seine. Block nets are set upstream and downstream ends of the reach. 2 probes are stretched across the stream. These have metal rods hanging in the water to deliver electricity. Electric seines run by a gas-powered generator. This stuns the fish. They float to the surface, and then they have 3 or 4 netters behind the seine to catch the fish. The fish then are deposited in an aerated tank. You can see in their boat for later to be ID'd after the survey is over. If the stream has high conductivity, the electricity does not work as well. The fish don't get stunned, so they have to use minnow seine nets instead. Minnow seine nets are stretched across the stream and then they're pulled upstream. And then the 2 ends of the net are pulled together and fish are laid out on the bank for ID. In the photo on the left, you can actually see some of the biologists are kicking up rocks to dislodge any smaller fish into the net, and then they'll bring it upstream and identify it. Lastly, they do boat electroshocking. This is used when the stream is too wide or deep for an electric seine. It's similar to the electric seine where it delivers electricity, but it uses 2 large booms that hang over the front end of the boat. And They typically sample for an hour from wherever they launch the boat if it's feasible. The boat shocking can miss smaller individuals, so seine hauls are also used to get smaller specimens, kind of like this. This is a juvenile gar up top. So if a fish can be identified at the stream, they will, and then it'll release it. But if the specimen is questionable, they'll then preserve it and then identify it at their lab. They record length and weight of all fish, over 4 or 6 inches, depending on species. Fish consumption analysis is only collected at selected sites. They fillet the fish and freeze it for later analysis. DNR verifies their data and they calculate their own fish IBI. And then when we're ready to conduct our assessments, we'll reach out and they'll provide us with their data. So most of DNR's identification is done at site, but EPA's sample processing is all done in the lab. So samples were preserved in ethanol on site and stored in the lab until they could be picked. We pick 300 organisms subsample for every site. Samples are rinsed and distributed evenly in a 30 grid tray. We have to pick at least 4 squares, and squares are chosen by a random number generator. If there's too much debris, we pick at least 4 squares, shown in this example here. Pick 4 squares, and then those squares are taken and distributed in a second tray shown on the left. And then that tray is used to pick the 300 organisms. And again, another four squares has to be picked. One thing to note on this, there's a dragonfly here. If it's crossing 2 squares, whichever square the head is in is the square where the dragonfly would be counted. So inverts are picked out of the debris. You can see here from a large tray, and then checked underneath a dissecting scope just to make sure they have a head and they are counted and placed in vials for the taxonomists. Typically we get 3 vials per sample. So a 300 count vial, a midge vial, and a unique vial. The unique vial is after the 300 count is picked, we sift through the remaining debris for anything that we didn't see previously for the 300 account. Also, larger specimens are collected just in case they're better for identification. Semi-aquatic insects are not included in the 300 count. And if you've been staring at the debris, I was curious if you found the caddisflies. There's at least 2 of them. They're right there. Kind of reminds me of I Spy. So then this is when I come into play. Macroinvertebrate identification. The EPA currently has 3 state taxonomists. 2 are located in Des Plaines, and myself during the off season. So starting October 15th or sooner, depends how the field season goes. I start identifying samples, either finishing up the previous field season or starting on the current season. We identified to the lowest possible taxonomic level using various dichotomous keys. Typically, we try to get to genus or species level. If a specimen's too damaged or missing identifying characters such as gills or mouth parts, or is too immature, we'll leave it at a higher level, such as family. If a specimen's missing a head, we discard it. Individuals are counted and recorded to be later entered in our database. Most individuals can be identified using a dissecting scope. This is an image of a typical sample. you can see in the glass dish. But the family Chironomidae, their character identifying characters are too small. We did a need to slide mount them on slides. We need to do this in order to get a clear view of their head shown in the picture in the middle. See their mandibles, their mouth parts, and antenna. The taxonomists meet annually to verify each other's identifications. If there isn't consensus, we reach out to other state taxonomists for verification. And since COVID, we haven't been able to meet in person, but we still share each other pictures to verify. And also since COVID, I currently have turned my kitchen into a lab, and it's been working out well, and my lab assistant is very helpful. So with the identifications, we can calculate a macrovertebrate index of biotic integrity, or MIBI. We don't include the unique organisms, and we also don't include the semi-aquatic organisms, which includes a lot of the hemipterans. So this is a picture of a water scorpion, and some beetles don't get included into the calculation. Others get standardized to a higher level, such as leeches, crayfish, and clams, and worms. All the inverts are assigned a functional feeding group and tolerance value, and those are used along with abundance to compute the MIBI with the 7 metric values that are shown in this table here. The samples are run through an access database IBI calculator. The metrics are transformed from a 0 to 100 using best known values. These metrics are then averaged, and that's how we get an IBI score for each sample. So this is a little overwhelming, but this just kind of sums up what we've discussed so far. So this is our water collection, EPA analysis, and the data is reviewed by a team and then enters into our water quality database. DNR, their fish surveys, their identification enters into their database. The habitat, our 20 jab collection, samples picked, identified, and entered into our invert database. So basically these 3 databases, water, fish, and macroinvertebrates, get inputted into our statistical program, and that is automated and produces suggested assessments, and then is reviewed by our biologists, and then goes into the US EPA attains database in our integrated reports. So here's a simplified version of that. So results from each individual analysis. So again, biological, water chemistry, and habitat data are evaluated together to determine if the stream is fully supporting or not supporting. In this example, I'm using aquatic life use. Biological data is given the most weight in the assessment. This is because lifespan of organisms represent an extended time frame versus a window in time like water chemistry. Water chemistry is considered second, especially if the biological indices are conflicting. That means the fish and the bugs are conflicting, and then the habitat is considered. So all these are inputted into our statistical program and produces a suggested assessment, whether it's fully supporting or not supporting. Then the biologist in each regional office looks at the suggested assessments, and with considering any additional site-specific knowledge, gets the final assessment, decides on the final assessment. So to wrap it up, the reason why we're doing this is to assess water quality for aquatic life use. aesthetic quality, recreational use, primary contact, and fish consumption. This is done with the ambient water quality program and the intensive basin survey program that's done on a 5-year rotation. We went over field work and lab work that includes site selection, recon, collection of water, inverts, habitat, and fish, a little bit about sample processing and data entry. For the assessments, the data is consolidated in R. Biology is weighted more than water chemistry and habitat. EPA biologists decide on the final assessment. And then these are all published in our integrated reports that includes what streams were assessed and which ones are impaired with violations. This also produces a 303D list. The 303D list includes the impaired waters, a list of violations, and causes. So this wraps up all the stuff that I was going to talk about for the intensive program. Just to make a couple notes of like we're working on other projects that we're working on now. We're working on consolidating our data. It's going to be a 2 to 3 year effort because we have multiple databases we're using right now. We're using one for macroinvertebrates, one for habitat, and one for water. We're hoping to combine all those and then to utilize US EPA has a program called How's My Waterway. This is an example of Minnesota. So you can go on the US EPA website and it's a easy access for the public to look at how your waterway is. You can zoom in on a watershed or a stream, and it gives you the different designations that you can look at to see how many streams were assessed and how many were impaired. So hopefully after our data gets consolidated, we can streamline and be able to publish our data on this website for easy access. We're also going to work on a diatom index project this summer. We're working with Indiana. They just came up with a diatom index. So we're just going to collect some diatoms at each site that we go to and ship them off to Indiana, and they're going to compare an Illinois index to Indiana. Also to be available is a harmful algal bloom dashboard. It's going to look a lot similar to this. It's actually a PFAS dashboard that is available now, but a similar one is in the works for harmful algal blooms. It's basically an interactive map with sample location and results. Results should be up within 7-14 days, just depends on the toxin. And they just recently added a expanded gallery on the harmful algal bloom site, which is here, which I can share all these links with Hannah, and she can share with everyone. And here's just some pictures from the gallery. And lastly, we also just are almost done with a lake macroinvertebrate IBI. We're one of few states that actually has a lake macroinvertebrate IBI, so that's pretty exciting. We recently had one, but we were processing our lake habitat samples separately, and now the IBI is based off of our composite sites or composite habitat samples. But more can, if you're interested in this, more can be found at this biomonitoring in Illinois lakes site, which again, I can share the link with Hannah. It's a rough draft right now, but it explains the whole process. And if you're just interested in the lake monitoring side of things, I talked about streams today, but if you're interested in lakes, you can also find that on the EPA webpage. And with that, I will take any questions.
Great, thank you. That was a great presentation. lots of methods, which I
Yeah.
like. I like seeing the different ways you can monitor water bodies. There is one question in the chat right now, which you kind of covered it already, but if you want to go over it again, Pam asks, what's a jab?
What's it? So a jab is a sample collected in a net. If I can. All right, can you see that?
Yeah.
So this is our net. We do 20 jabs to represent a site. So if I need to take, if my bank zone is 8 and I need to take 4 samples in wood and 4 samples in roots, I would go sample a root and that would be considered 1 jab. Does that make sense?
Yeah, so you're basically scraping the net against the substrate, right?
Yes.
Okay, yes, that makes sense to me.
I do it so often, I don't know, it's hard to
Yeah.
explain. Yeah, one jab represents one habitat sample. And that was the different, like the bottom zone and the bank zone I was talking about.
Okay, we have a question from Danielle, and she said that she really enjoyed the presentation. And she says, I noticed that in the Little Wabash watershed, there weren't many or any sites in the major branches to the east or west of the main channel. Obviously, you can't sample everywhere, but is there a reason for this?
Let me go back to my map. Okay. Is she talking about these? I'm assuming. So this is the Little Wabash. This is Skillet Fork, and that gets sampled a different year. And this is Middle Wabash or Wabash. So those are sampled on a different year, if that's what she's referring to. So this year we're just focusing on the direct trips to the Little Wabash.
Danielle, is that what you were asking about?
Yeah, and I was just trying to type it instead, but yeah, I thought that like that watershed boundary was the whole area that was supposed to be sampled for this year, not just part of it. So
no, that you're right, that is a little misleading, but yes, this is the Skillet Fork and this is the direct tribs to the Wabash. Those get sampled on different years.
Great, thank you. And then we have a question from Kathy that says, what is the craziest thing you've ever seen or collected on a site?
Oh, man. Well, we do see a lot of trash. I excluded any of those pictures just because they're not pleasant.
Mm-hmm.
But I have seen like a full vanity set, and it was almost like set upright in the stream. It was just very out of place. There was a sink included. It was like, why is this here?
Yeah.
But I like collecting those large megalopterans too. I get very excited when there's one as big as my hand.
Those are the ones I don't like collecting.
Yeah. So invert wise, I like when I get like the large individuals.
Mm-hmm.
But yeah, craziest thing was just that, I don't know, the random trash you see in a stream, which is unfortunate, but.
Kathy also asked what you like best and what you like least about your job.
I'm a bug person through and through. So anything related to the bugs, whether it's sampling them or identifying them. I love getting new caddisflies. I'm a big caddisfly person. What was the second part of that?
What you like least.
Oh, probably the heat.
Yeah.
Sometimes I can't handle it in the waders on those above 90-degree days.
Yeah. Jim asked if any of the RiverWatch sites are also sampled by EPA, and do you use any of the RiverWatch data?
I am not sure if we sample the RiverWatch sites. I don't know. Do you have set sites? I guess I'm not that familiar.
I don't think we've ever looked at our sites and compared it to EPA sites, so I'm not sure on the answer to that either.
Yeah, I don't know.
Okay, well, it's something we can look into.
Okay.
And then Steve asked, in smaller streams, depth in any given place can vary greatly from year to year. How does this affect your work?
Well, that's why we try to sample at base flow conditions. There are a lot of streams, especially like sand and small gravel streams, that shift a lot. So maybe there's a lot of, once there was a pool here, but now it's filled up with sand and the deep pool's on the other side. I don't know if that's what they're getting at.
Do you ever come to a site and realize like it's too low to sample?
That only happens with their third round water. If we know the stream looks like it's going to drop out, we make sure to do that in June. We adjust our sampling. So if it looks like it could dry up, especially like some of, maybe the smaller Shawnee stream, we try to sample those in June and try to collect our water quicker before it dries up.
Does anybody else have any questions for Jodi? Michael asked, do you count any invasive species?
Yeah, if I get them in the sample, I count them.
Do you guys ever look for them while you're out sampling? Like, oh, we saw some zebra mussels while you're sampling or anything like that?
I mean, if they're in the sample, they will be, part of the sample. They'll get included in the 300 count, and that plays effect with the macroinvertebrate IBI. But I don't see a whole lot of invasive. The only thing we get a lot of is Asian clams. That's our main invasive that we get. The Illinois does have the rusty crayfish, but that's more northeastern Illinois, and they're not down here, so I don't see them.
Okay. Okay, well, if nobody else has any questions, thank you, Jodi, for your presentation, and thank you everybody for joining us tonight. This is the last speaker we have for this winter speaker series, but we'll be back in the fall with more presentations. And I just want to remind everybody that our RiverWatch training workshops are coming up. So if you're interested in attending one, you can find them on our Facebook page, and you can also find them on the events calendar on our website. Thank you all for coming, and have a good night.